Good morning. This is Dr. Rajalakshmi and Brandon, Head and Assistant Professor of Biochemistry Departments. Today we are going to discuss about enzymes. So the enzymes, it's a catalyst. So the term of enzymes, coined in 1877, German physiologist Enxymes coined the word from the Greek word enzymon. Its meaning is yeast. Enzyme as biological agents in 1857. Louis Pasteur established the fermentation that was caused by the living yeast cells. Enzymes are biological catalysts. They accelerate biochemical reactions without being consumed. Most of the enzymes are proteins in nature. Enzymes are highly specific in their actions. There are two types of enzymes, which is endoenzymes, intracellular enzymes, which is present inside of the cellular, and then exoenzyme, that means extracellular enzymes. Endoenzymes locations present inside of the cell and greater than. And the primary role of endoenzymes is cellular metabolism, energy production, and synthesis of cell components. So the small molecules that have already entered into that cell, that is endoenzymes. So for examples of endoenzymes, enzymes involved in glycolysis, Krebs cycles, and DNA replications. Say for example, DNA polymerase. And exoenzymes, which is present outside of the cells to functions externally. The major role of this exoenzymes breaking down large complex polymers into a small nutrients. Substrate large macromolecules like proteins or starches outside of the cells. For examples of exoenzymes, so digestive enzymes analyze amylase, pepsin, and bacterial toxins that collagenase. And next we are going to see a nomenclature of enzymes. International Unit of Biochemistry (IUP). Enzymes are named according to the reactions and their catalysts. So most of the enzymes names end with ASE, which means each enzyme is assigned as enzyme commission number, that is EC number. For examples, lactate dehydrogenase. So the classifications of enzymes based on IUP. Enzymes are classified into 6 major classes. The first one is oxidoreductase, transferase, hydrolysis, lyase, isomerase, and ligases. So oxidoreductase, that is EC number 1, catalyze oxidations mechanisms, reductions mechanisms. Examples, lactate dehydrogenase, and the reactions lactate plus NAD plus equal to, it gives pyruvate plus NADH. So transferase, that is EC2, that functions transfer the functional groups from one molecules to other molecules. For examples of transferase, hexokinase and aminotransferase. Okay, the third one is hydrolysis, that is EC3. That function is catalyze hydrolysis reactions. For examples of hydrolysis, amylase, lipase, trypsins. The fourth one is lyases, EC4 functions catalyze additions or removal of groups without hydrolysis. For examples, fumarase and aldolases. So fifth one is EC number 5, isomerase, that function catalyze intramolecular rearrangements. That is phosphoglucose isomerase racinase. So the sixth one is EC number 6, that is ligases, join two molecules using ATP energy. For examples, DNA ligase, pyruvate decarboxylase. So as we discussed about so far, that enzyme as catalyst. What is the main characteristics of enzymes? That increase the rate of reactions. We might add change after the reaction also, because it's required only small amounts and it's highly specific, lower activations energy. So what is meant by activation energy? Activation energy is minimum amount of energy which is required to initiate chemical reactions or biochemical reactions. What is the main role of enzymes in this reaction? Enzyme lower activation energy, this increase the rate of biochemical reactions, the equilibrium of reactions remains unchanged. So, enzymes we discussed, this is biological catalyst. The IUB classifies enzyme into 6 major classes. Enzymes was named according to the reactions that catalyze. Enzymes lower activations, energy and increase the rate of rates. Enzymes essentially for all metabolic process. Then we are going to see the enzyme specificity. What is enzyme specificity? A specificity refers to ability of enzymes to recognize a particular substrate and that catalyze a specific reaction. This specificity is due to unique three-dimensional structure of enzyme active site. So there is a type of specificity. Number one is absolute specificity. What absolute specificity happening? The enzyme acts only on single substrate and catalyze only one type of reactions. For examples, urease acts only on urea. And then second one is group specificity. An enzyme substrates containing a particular functional group irrespectively and then remaining the cellular structures. For examples, hexokinase and then linkage, that is bond specificity. It's otherwise called as bond specificity. The enzymes acts only on specific type of chemical bonds regardless of its molecular structures. For examples, triglycerides plus water molecules gives fatty acid plus glycerols. The fourth one is stereospecificity. An enzyme acts only on stereoisomers of substrates and not in its mirror image. For examples, L means levorotatory and D means dexorotatory. L-amino acid acts only on L amino acids and D amino acids acts only on D amino acids. Hexokinase acts on D-glucose but not on L-glucose. What is the importance of this stereospecificity? Enzyme difference between differential spatial arrangements of molecules. That is optical specificity. The most tricky aspect of specificity enzymes that particular enzymes will react with only two optical isomers. One of the two optical isomers. For examples, arginines acts only on L-arginine and not in its D isomer. Similarly, D amino acid acts on D amino acids only the corresponding ketose acids. So next one is geometrical specificity. Some enzymes exhibit specificity towards the cis-trans forms of examples. Fumarics catalyze the interconversions of fluoric and malic acids. So what is then next we are going to see the active site of specific three-dimensional region of enzymes. Where the substrate binding the chemical reactions takes place, it's formed by two amino acid residues that provide suitable environment for catalysis. So characteristics of active site. What are all the characteristics active sites which is present in the enzyme? It occupies a small portion of enzyme molecule, shape complementary to the substrate, and contains catalytic group that participate in the reactions, provide a specificity to the enzymes, forms temporarily enzyme substrate complex. For example, for the active site, in the enzyme sucrase, the active site specifically binds with sucrose and catalyzes its hydrolyzed into the glucose and fructose. So, for active site, there are two hypotheses. One is lock and key hypothesis, then another one is induced field triad. So, now we are going to discuss about lock and key hypothesis. It was proposed by Emil Fischer in 1894. According to the lock and key hypothesis, the active site of enzyme is rigid and has fixed shape. Only a substrate with a complementary shape had fit into active site. Just a specific key enzymes in the particular lock, and this enzyme substrate gives enzyme substrate complex. Finally, it gives enzyme and product. The E represents is altered enzyme conformations. So this is the lock and key model. What are the features of active site? It's flexible and substrate binding induce a change in the enzyme shapes, explains higher catabolic efficiency and broader substrate specificity. And for example, hexokinase change its shapes when glucose binds to its active site. So next one is induced fit theory, which was proposed by the enzyme active site change shapes to accommodate the substrate, making it the most widely accepted model for enzyme actions. And thank you for your participations, and thank you all.